Hello and welcome. It's my great pleasure today to introduce Dr. Andres Moreno Estrada. He received his MD at the University of Guadalajara and then his PhD at the Institute for Evolutionary Biology in Barcelona. Two lovely places. He then went on to do his postdoctoral work with Carlos Bustamante, first at Cornell and then at Stanford. And for the last 3 years has been the director of genomics and the core labs at Langevio in Guanajuato. Okay, so today he's going to tell us about some of his research projects in Latin America. And welcome, thank you.
Thank you. I think I might have my own mic.
You have your own mic.
All right. Thank you, Anne, for the introduction and mostly for the invitation, and every one of you for coming today to hear about our work. So it's really a pleasure to be in such a great place and a stimulating environment with all the people that have been meeting throughout the day, and I'm looking forward to the meetings that are next, the seminars. So it's really a pleasure to reconnect and put some faces to really famous names, as many of you are. So as Anne was mentioning, today I will be talking about these broad spectrum of research lines that we have in our newly created lab that mostly refer to Latin American diversity and more recently about the Pacific. So I'll try to cover both of them to give you a flavor of what kind of research we're doing currently. So just to recap what Anne was mentioning in her introduction, I would like to start by telling you a little bit about myself. So I'm originally from Mexico. This is my hometown, Guadalajara, and this is the beautiful building of the University of Guadalajara where I study medicine. So yes, I'm an MD, but I have never practiced anything about clinics. So don't ask me if you have anything, just find your doctor. Then I move on to do, to become a true doctor in something, which is a PhD. And believe it or not, I managed to get a PhD out of this beautiful and distracting place where we always debate between, going back and writing a thesis or just relax at the beach and play some volleyball. But yeah, it's a great place to do research. They have this nice, beautiful research building only for biomedicine research. So it's really a great place to be. And then after that, I moved to this other beautiful place closer to where we are right here. This is Stanford University in California, where I spent 5-6 years doing my postdoc, and then I moved to a research associate position before I took this other faculty position that is the one that I have now in Mexico. So just for those of you who don't know where Guanajuato is, this state in the right center part of the country, and right there we have this beautiful campus that seems like from other planet, because if you go out there, it's like mostly crops and not very attractive looking place. But then you find this nice building that Maria can testify that is really a nice place to be to do research. So mostly we are a plant genomics institution. So mostly was developed because a neighboring campus was already doing a lot of genetic engineering in plants. So now it grew to be one of the major genomic facilities nationwide. So its research lines have been growing towards more a global spectrum of genomics. So not only plants, but also animals, and now we're doing also human genomics. So yeah, we have two grad programs. One is called Integrative Biology, to attract people that is interested not only in plants, but again, more in a broad spectrum of biology and combining genomic tools with bioinformatics and computational power. So I'm also the director of, this is actually our recently created lab. So it's a small size lab, but I'm really happy to have really talented people joining the lab in the past 3 years. So now we reached the maximum magic number of 10 people in the lab. So I'm really happy for that. And this is what you see in the above picture is basically the space that we have for students and people working on the left side. Part of the picture you see our lab, and then the entrance is where people is at that point looking at a exhibition of ancient skulls from an anthropology exhibition in Mexico. So it's a nice place, a nice environment to work with. Like I was saying, I'm also the director of the genomics core facility. So in this place, I lead another team that it's about 10 technicians working towards offering service not only locally for researchers in the campus, but also external users nationwide. And as some of you are, also external users from abroad, other countries like the US, Germany, Peru, and Chile. We have been receiving and processing samples from all these collaborators, and I'm happy to show you maybe more about what kind of services we offer. You can visit the webpage right here. It's LabSerGen for Laboratorio de Servicios Genómicos.lanhebio.sinvestab.mx. Okay, so basically that's what we do. Research and provide service of platforms to enable people to do genomics in the region. And I don't need to convince this audience actually to explain that the reason why we are in this field offering genomic data to all sorts of different research labs is thanks to this explosion of genomic platforms that have been growing in the past 10, 15 years and whose prices have actually been going down also in the past 10, 15 years. So thanks to that, we all know that there has been an availability, an increasing availability of genomic data from all sorts of species, and that's one of the major interests of our institution, which is a biodiversity institution. By the way, did I say that LANHEBIO stands for National Laboratory of Genomics for Biodiversity? So that's LANHEBIO, aka Launch Bio, as we like to refer to it in English pronunciation mode. So yeah, for LANHEBIO, it's really important to have data available from all sorts of organisms. But when it comes to humans, that's our topic of interest, there's a big problem, at least in our field that we're interested in exploring understudied populations. It is important to know that there's a tremendous bias towards European descent population that have been mostly participating in these genomic studies in the past few years. And this is a bias that has been ameliorating. So actually the problem is less severe as it was, for example, in 2009, where up to 4% of the participants, or only 4% of the participants, were of non-European ancestry. If we take a picture of 2016, the bias is less severe, but it's still there. So we still have up to 81% of the participants in GWAS studies coming from European descent population. So the rest of the diversity of the planet actually gets squeezed into this 4% or 19%. And within that little chunk, actually, this big part is of only Asian descent population. So if you think about the rest of the diversity in other parts of the world, like Africa, like the Americas, is still missing from that part. So that's kind of the motivation for us to really focus on these populations. But why does it matter for medicine, for example, right? I mean, and this is coming from an MD that tells you that doctors really don't have a clue most of the times. Sometimes they do. And let's take an example of here. You have a patient that says, well, I have this problem. The MD, the physician says, well, it's all in your genome, right? I mean, what are we going to do? So basically like, well, it's all your fault, right? You have bad genes or whatever. But reality, if the clinical world don't really pay attention and don't understand where these variants that actually do have a clinical consequence, they don't understand where these variants are coming from, it's going to be difficult for us to distribute these clinical efforts based on research, of course, of these particular populations. So we have to really have an evolutionary point of view to really make a sense of the clinical consequence of variants that have been evolving over time in our species. So, and of course, when I mentioned that we have then to take into account the evolutionary history of our own species, and this is a map that I'm sure all of you are familiar with, but this is just to mention that we all know that there are many evidences pointing towards African and recent origin of our species, and that still is by far the deepest genetic signature that we can find in genomes of human populations. And out of Africa, then we start having these human dispersals that also left different signatures in populations that have been differentiating over the past 100,000 years or so. And when I say that we can actually reflect this in genetic patterns, I'm going to refer just maybe to the most recent effort towards that. Not the only one. There's many, many previous efforts, and they back to the 90s. Like for example, Cavalli's Forces project in the AGDP actually started at Stanford with Mark Feldman and with people all over the world. But again, just to give an example of one kind of approaches, this is 1,000 genomes populations with genome-wide data showing the proportion in this graph here particularly, the proportion of novel SNPs discovered by each population group. And you have by color different continents, like for example, all African populations are in these yellow shades, for example, Europeans in blue. And look at the Americas populations that for the first time in a genome-wide study were included Latino populations. Let me specify that these were not indigenous populations from the Americas, rather cosmopolitan samples from capitals. Like Colombia, it's like Medellin from Colombia, that the CLM, or PEL stands for Peruvians from Lima, and so on. So we do expect to have a mixed pattern from those. What I want to highlight here is that, as expected from evolutionary history, we see that the highest numbers of novel variants are found in African populations. And then you can see that all the rest of the world is actually falling in this average number between 4 and 4.2 million variants. But look at the Americas samples, actually are the only ones spreading between the two, right? Like some of the individuals from these populations have actually similar counts of novel variants as the rest of the world, like Europeans, Asians, and so on. But there are some of them that actually have similar counts that are as high as the Africans. If you look at the particular populations, we see ASW, which is African Americans. Not surprisingly, they have a lot of African ancestry. ACB from Barbados. But look at these, for example, CLM, right? Or PUR, which is from Puerto Rico. You have some individuals there that also approach very similar patterns as Africans. And not surprisingly, again, this is due to history and this process called admixture, right? So this whole history about human evolution changed dramatically in the past 500 years because we have this collision of previously differentiated populations that now coexist and start to mix. And we know that that has a very clear impact in the genetic pattern of populations that we saw from the 1,000 genomes graph. But it also has an impact in the phenotype, right, as we can all see here. And you can here pick your favorite character and already tell, well, which one has more African ancestry or European ancestry and so on and so forth. But genetically, we also can tell just from a basic analysis like PCA, for example, which ones have more, a higher proportion of a particular ancestry. So this is an example of some Latino populations from the Caribbean and neighboring countries, like, for example, Cubans, Dominicans, Puerto Ricans in green. And as you can see, they actually fall intermediate between this spectrum of African ancestry and European ancestry there, where others like Colombians and Honduras have more Native American ancestry. Okay, this was like a very small sampling study, again, with cosmopolitan samples from these countries. But here we still fall short, I think, in this simplistic view that Latinos have this discrete pattern. Like, okay, either if you're Caribbean, you're going to fall in this section of the graph, or if you are maybe from the mainland, you're going to be more Native American, and so on and so forth. But let me show you an example of a bigger study with bigger sampling where they had actually people from all over the world. So this is called the PAGE study, and this is a big consortium genotyping more than 150,000 samples all over the world. And as you can see, there are samples again from all continents, right? And you have the reference samples colored, like the Americas in blue, Europeans in green, and so on. But now in gray, you have people from other non-reference samples, okay? And let me just highlight where the Latino populations are falling in this PCA spectrum. Okay, they're basically all over the PCA space, right? So not as discrete clusters as before, like, categorizing if you're Caribbean or not, etc. So this is just to give you a flavor of how big is the spectrum of diversity when you classify populations such as Latino, right? And that may have different connotations depending on how you take that concept. But in genetics, it has a really broad spectrum of what could it mean, right? So try to link this back to the evolutionary history of each population and the clinical consequence of that is kind of what we're doing. So let me also give you a background that we're particularly interested in not only in these big continental scale patterns of you're more Native American or more African or more European, but particularly in this corner of the PCA space, we are collapsing all the diversity of the American continent as just one ancestry component, right? So but in reality, we know that that's not the case. There's also a lot of diversity within the continent, and we're trying to figure out if we can disentangle these components in a finer scale. So that's what has been motivating our research and many of others before us. We're not, by far we're not the only ones interested in this. This is one of the seminal works by one of the major players in this field of the Americas population genetics, like good friend Colombian researcher Andres Ruiz Linares and his group generated this data set, which is one of the first genome-wide data sets, not based on SNP array dense data yet, but STR. And we're able to see this pattern that actually heterozygosity changes geographically within the Americas. As you move south, you see a drop in diversity that is lower and lower as you move away from a point of origin in the Beringian area, and then that's the point of entry from East Asia into the Americas. So again, in agreement with what we know from history in the colonization of the continent. But that's, again, a view that was even proposed a long time ago, not with genetic data, but also with linguistics. And a further study based on SNP array data now with the same data set basically confirmed the idea that, for example, Greenberg proposed about separating all Native Americans in at least 3 major groups, based on linguistics, of course. And this was the famous Amerindian group, which was the majority of Latin America, of what nowadays is Latin America, Eskimo, and the Nadene speakers as the other 2 groups. So basically, genetics was also kind of reflecting this same major partitioning. But this is still, for us, falls short in the full spectrum of diversity that we know that even within Latin America, there's many different sources of Native American people. So taking again this example of the same study with these 52 set of populations from all over the Americas, look at how when you cluster them in an admixture plot, they actually fall all in this same blue ancestry component. Of course, at this very basic level. You can partition that into subcomponents, but at least at the continent level, only the ones that are at the northernmost part of the continent, they have some mixture with this other green Siberian component. So there's still this majority of populations from Mesoamerica southwards that share this same ancestry component. Again, in agreement with a single source of all these populations, but also this vision lacks the specificity of the substructure in that component. Again, wondering if that could be recapitulated knowing that there's a lot of diversity. So that's how our working group in general, our extended collaborative group, has been focusing in the past years to do more like regional approaches to understand the diversity of regions like, for example, Mexico or the Caribbean or South America. And I'm going to give you very few examples of these 3 regions and talking about what we're currently doing. So, for example, back in 2014, when we were doing the Mexico Diversity Project, this was mostly done in collaboration with many people that in Mexico has been already doing a lot of collaborations and long-standing relationships with local communities and anthropologists, and we were able to put together a set of nearly 500 samples and genotype them with the AFI 6.0 array, which is almost a 1 million SNP array. So with this data set, we were able to create this graph that basically is based in genetic similarities between individuals, where each node is one individual, and the lines connecting them tells you if they share more than 13 centimorgans, just to put a threshold between these 2 individuals. So as you can see, the first pattern that emerges is that most of the connections are actually within the populations, right? And very few lines connect individuals from different populations. Speaking about mostly a history of isolation and very few genetic flow between populations, with some exceptions of, for example, like outbred populations like the Mayans, for example, that can be found some connection with distant populations in the other side of the Gulf of Mexico, or nearby populations. For example, these 3 are Nahua speakers, all of 3. This is not geographically at scale. This is just like a reference. I mean, this guy is not really drifting into the ocean. This is just like, just to have a close connection or to where more or less the sampling location is, but this is not exactly a coordinate-based map. But it gives you an idea of how isolated these populations pre-contact were. So now talking about this complex pattern after contact, and again, because we know there's a mixture, we now have to take into account that individuals can be partitioned in these ancestry proportions. And this is just a basic analysis like admixture does, for example. And I don't need to remind you how this works. I mean, each individual can be allocated to different membership to this ancestry component. Imagining, for example, this is just a 3-way admixture model. Individual A can have this much of population 1, and then a little tiny population 2, and the rest of the genome coming from population 3, and so on and so forth. We can see the different patterns across individuals. So we wanted to do that in Mexico, in the cosmopolitan areas, not in the Native American groups. So to do that, we started a collaboration with this big National Institute of Genomic Medicine in Mexico, who was at the time precisely doing a lot of sampling efforts in cosmopolitan areas. So I'm highlighting the states in which they collected samples from capitals. So for example, from Sonora, these were samples from Hermosillo, and so on and so forth. So we wanted then to combine this data with our data set from mostly rural and indigenous communities, and we ended up having this data set. So on the left part, you see the references, which is Africans in green, Europeans in red, and then at the top, you only see the Native Americans in blue at the very basic model of three-way admixture. Then when we allow the model to infer 9 clusters of ancestry, we can see that there's substructure both within Europe, which is a north-south line that we already know about, and look at this heavy substructure pattern that we see within the Native Americans in Mexico. So you have components that, for example, are private to some populations, like the Seri in the desert, in the Sonora desert. It's a component that is basically shared by nobody else. It's with this navy blue that you see that almost nobody else except for the northern ones have a little bit more. Then there's these more spread components, like the one that we call the northern indigenous component, represented by the Tarahumara, the Tepehuano, and so on. Then you have this other, this is actually the major component everywhere in Mexico, which is kind of the southern component, mostly represented by people in Oaxaca. So like the Zapotec, like the Mije, and so on. Then you have this other regional component that we call the Mayan component that is also shared, interestingly, not only among Mayan speakers, but we also find some connection with the eastern coast of Mexico. Again, maybe supporting these connections with the genetic similarity, the IBD map that I showed before, maybe reflecting that connection as well. But now let's look at the admixed cosmopolitan samples in Mexico, which, just as a reminder, they're from these states. And they, of course, have way more of this red component that is European mixture. But if you focus your eyes only on the blue side, which is basically taking from the top panel, just focus on the blue component, and then you go to the K9 bottom, you see that they actually follow more or less a similar pattern as the substructure we see in the Native Americans. So for example, Sonora, which is a northern state, is the only one that has this high proportion of the native blue northern component, which comes from here, and so on and so forth. The Yucatan, for example, is the only state that has this large proportion of the Mayan component. So what we are seeing is that the mixed cosmopolitan samples still retain a lot of the substructure of the Native American groups. So to study that further, we developed this algorithm that actually is an ancestry-specific approach for many of the regular analysis that you have heard in population genetics, like PCA, like FST. So we implemented first this simple analysis with admixture. So you start with genomes that are admixed from these 3 sources. Then we phase them, and we apply this local ancestry algorithm to infer which part of each chromosome comes from which of these 3 continental sources. Then what we do is a second round of PCA, now with a larger set of population from that particular continent. For example, if we want to infer the origin of European blocks of a mixed individual, we only take those coordinates, we mask everything else, and combine with a larger set of European populations. And we can do the same for the African component and the Native American one. So what I'm showing you next is basically the ASPCA of the Native American blocks of the admixed Mexicans, okay? So surprisingly, what we found, instead of that triangle-shaped PCA, remember in the beginning that everyone collapses in one single point, whether you are more Native American or more European, we know that these guys are mixed. But if we take only their Native American fraction of the genome, they're no longer collapsing in a single point, because we are allowing them then to project on a bigger panel of Native Americans within Mexico. And what we found is that there's actually some dispersion. These dots in blue are far away from this other in orange. And when we map back where these samples are coming from, and these are the states where they're coming from, you actually see that they recapitulate a geographic map of Mexico. Just like, for example, the famous paper by John November in Europe, right? That you could do straightforward with that masking, because in Europe there's more homogeneous pattern without recent admixture as in Mexico. But in Mexico, we needed to have this specific pipeline to uncover this signature. So that was nice, but we wanted also to see what this has to do with biomedicine, right? So we had access to another data set that has measurements for pulmonary function. And I don't have time to go into the details of the correlation we did, but this is a summary of different states of these sampling individuals, right, that have the predicted change in this measure of lung function, which is FEV1, basically assuming that you fall in those states, you can then now follow or extrapolate what is the differentiation based on your ASPCA values. If you're from different states, what's going to be your predicted lung function value? And as you can see, it really changes a lot if you are from Sonora, for example, than if you are from Yucatan. A total of approximately 7% change is predicted if you live in Yucatan or in Sonora based on just the different type of Native American ancestry you have. So even though this was not a direct measure of phenotypes in these individuals, this was just, like I said, an extrapolation from another cohort, this tells us that this might be important for medicine in general. I mean, the substructure of Mexico might be important for many other traits that are relevant to medicine. So with that motivation, well, by the way, this was published in Science 2014. We never got the cover of the magazine, but that's exactly why I want to show it every time I can. Because I like the figure that we proposed to Science as the cover. So this is it. And after this project, our motivation has precisely to see if the same substructure pattern has an effect in additional phenotypes. So what we're doing right now in the lab is that we're genotyping a big biobank, which is called the National Health Survey of the 2000. This is a time where the Mexican government was collecting data for demographics and health purposes, but also they included blood sample in all 40,000 individuals. So this is really a true valuable collection of samples that were already extracted into DNA. And so far, there has been no genome-wide approach to study this collection of samples. So we proposed something genome-wide. Luckily, we got funded to do it from the Mexican government, but also from the Newton Fund in the UK. And right now, we're genotyping this small fraction of the biobank, not the whole 40,000, but at least we're initiating with the first batch of 10,000 samples that we're genotyping them in our core facility lab as we speak right now. So we're really happy with that. We're really excited to see what we can see in terms of ancestry components, but also in the correlation with phenotypes. Additionally, we're trying to also do measurements of 20 pathogens to see the measurements of antibody to these pathogens, to see if there's any correlation between the genotype and the immune response in different regions within Mexico. Because luckily, there's also serum samples, not only DNA samples from the same individual. So that's also one of the advantages of that. One of the main purposes of this lab is to generate a database that can be useful for many projects in the future, because again, this is really something valuable that costs a lot of money. It's really rare that in Mexico you have $1 million grant that is awarded to do something like this. So the idea is to be useful for many years to come. And I put as an example this other cohort in the UK that is, you may remember, it comes from this common set of control cases that they used several times in different papers. You may remember this figure from the initial GWASs that were able to discover hundreds of variants associated to 15 different diseases in a single study because they shared the cases in the same study. So you only have to genotype now. Sorry, did I say cases? I mean the shared controls, I'm sorry. So you only have to genotype your cases of each of the particular diseases. So you can reduce the cost of future UAS if you use controls available from that population. So that's one of the goals that we're trying to do with the biobank. The other goal is to build capacity. So like I said, we're doing this all locally in our facility. Up there we have the actual biobank where the samples are stored. This is the scanner in our core lab and the people involved doing all the bioinformatic analysis. This is also meant to be for local training and people to get familiar with genomic analysis. So with the ultimate goal, of course, of building capacity through big data projects, but that research also will inform back the community and decision makers to have health policies towards the community and the country. So that's kind of the ultimate goal. So moving on to other regions, I want to tell you very briefly that we also have been interested in working in these same patterns in the Caribbean. We have done work in the past trying to find out the origins of the Taino component in the Caribbean. And this was, again, a small study based on these islands, mainly from the from the Great Antilles, combined again with reference panels from different continents. And what we found there, following our ancestry-specific approach, is that we were able to tell that the origin of the Native American component in big islands, like, for example, Cuba, Dominican Republic, Puerto Rico, and so on, actually trace back their origins to northern South America, like an Amazonian component that is also consistent with what archaeologists and linguistics have been proposing. So with that approach, which is actually consistent also with linguistics, this expansion of the Arawakan-speaking people from the Amazon into the Caribbean 2.5 thousand years ago, it's nice to see that actually this pipeline can be applied to the region that have these long-lasting questions. But again, this is a simplistic view of the Caribbean because, as Maria Nieves pointed to me, just like Well, I have really a lot of interest to fill in these gaps that nobody has looked at before. Many other studies like ours and 1,000 Genomes have been focusing on these large islands. And what about the lesser Antilles, right? So this was very exciting to me because we also have never explored this, and I'm grateful that we started this collaboration through Maria with Adstone and Jed as well, who had been collecting this good data to apply the pipeline that we have put in place to uncover what the origins of these people is. And Maria did some preliminary analysis, and as in contrast with the other Caribbean islands, we see that these lesser Antilles individuals are mostly of African origin. So as opposed to being a mixture of this Native American European trend that we see in the other islands. So the idea here will be to apply this pipeline to uncover more what are the African origins of these individuals within West Africa, and that's what we are working on right now. So now let's go to South America, and I want to start saying that the striking thing here is that even though, again, remember the first picture of us sharing a big component on the continental level, if we do an ASPCA of different cosmopolitan areas of these countries in South America and mix them with reference panels, which are the dots, first thing we see is that there's no overlap of the genetic signature of these mixed individuals from different parts of South America with the Mesoamerican PCA space. So only at these two levels of North America, South America level, we see that countries within South America have a very, very different profile than Mesoamerica. So it's very important then to focus on particular regional approaches as well in South America, right? You see, for example, Peruvians here that they cluster mostly around the Andean reference population, and they don't even overlap either with Colombians, which have more this Amazonian-related ancestry component, right? You have countries like Chile and Argentina have a much broader space coverage because also their ancestors are much more richer in terms of origin. So that was also interesting to see, and we got more and more interested about South America. One of the examples is actually one of other Maria's projects here in the audience, and she just had this idea of writing for a postdoctoral position in my lab, and we got really lucky that she got an SF fellowship to join both Anne's and our labs in Mexico. So one of her projects is precisely to understand the genetic basis of the adaptation to high altitude, specifically to individuals who have been suffering preeclampsia. So Peru has a higher rate of preeclampsia in those individuals that have been moved to high altitude places without having Native American ancestry. So we have really way higher rates than other regions, other local regions that have been mostly adapted to that environment for thousands of years. So our idea is to find the genetic basis of that. And this has been really a tough task over the past 4 years. So we have been collecting the samples since 2012, and it's been taking us really a long time because cases are not really, even though prevalence is higher, they don't, walk into the clinic, every Sunday like 15 cases, right? So you have to wait until they arrive, and then these are carefully characterized and so on. So we have a collection of nearly 1,000 samples so far. So the idea is to genotype those soon. So Maria just went back to Peru. She spent a whole month there extracting this last batch of blood samples, so we hope to generate data and tell you more, maybe next year. So that's the idea on this project. But another project we have in South America as well is through this idea of understanding the regional patterns in particular countries, just like we did in Mexico, for example, is this the so-called Chile Genomico project, which is a diversity project within Chile, and sampling locations in different departments, as I call them. So the idea is to see also if there are different substructure components that we can correlate with history and clinical features. But through that collaboration, we were also interested in saying, hey, isn't Easter Island also part of Chile? Well, yes, it is, but normally Chilean government initiatives don't want to really get involved with indigenous population matters because they don't have really good relationship that explains why also, Easter Island was kind of non-voluntarily annexed to the Chilean territory not so long ago, so they don't really feel Chilean. And if you look at their culture, linguistics, and genetic patterns, they also do not look Chilean, right? So we were proposing, okay, why don't we visit the island and try to propose a diversity project to understand their mixture patterns? And that was back in 2013, where we were able to actually go to the island and do a sampling. So briefly mentioning where Easter Island is basically the most remote inhabited location on Earth. It actually has a very similar pattern in terms of admixture dynamics as in Latin America, because it also has the same mixture between different continental contributions, like, for example, starting with the Polynesian expansion. That's kind of the native component from the region. But also, we see, just like as in other colonized regions in the world, Europeans coming in and mixing with Polynesians in the 18th century. And then, since it is part of Chilean territory, there's also migration from the Chilean continent, so bringing some Native American haplotypes as well. So we said, well, this is the perfect setup to apply the same pipeline that we have been developing for other projects. And like I said, we run this pipeline to see how the population looked like. So we had all sorts of examples, right? So we had surprisingly some individuals that are very Polynesian, despite the fact that many people say, oh, no, there's no Polynesians left in Easter Island. They vanished like a few hundred years. So no, there are still some individuals that, of course, showed some mixture, but mostly Polynesian. There's also, some other people like this French teacher that wanted to really, she was very eager to be part of this study, and she was like, I want to know how Polynesian I am. I'm like, well, I can tell you right away, It's like, you're like the French teacher, it's like mostly European for sure, but there she is. And then most of the individuals look more like a mixture, like we have that example up there. One of the interesting part of this study was that participants were very proud of participating and knowing about their ancestry and their history, because they're very proud about their roots, their Polynesian roots, so they really wanted to know if that could be confirmed through genetics. So one of the things we did is to go back to the island after the sampling and actually return the results back to the community one by one, giving them this kind of genetic picture, as I like to refer to it. It's not a number, it's not a percentage, because we wanted to be very careful and not having them comparing their results, like, oh, I'm more Polynesian than you, right? So it's just your picture, as if you take a picture of you, but genetically, right? So this was fun, because they really got the idea immediately. If you see a lot of green segments, they were really happy. Oh, I'm very Polynesian, I knew it. So I'm going to go with the Chilean government and say that I'm not from Chile, right? So I'm Polynesian. So I don't know. We sparked the independence of Easter Island ever since, but so we're going back these years to see what happened. But the idea is to now go back to the lab and continue the research, because the idea is now more, this is more pictures from the sampling. The idea is to, what are the origins, genetically speaking, of these individuals? It's kind of a mystery, because they all know the culture is Polynesian. Clearly, they come from this Austronesian expansion from Taiwan. But which specific island gave rise to the original settlers of the island is kind of a mystery. So this is all data from what was available back then when we collected the first individuals. This is the Wolf General dataset, a project by Mark Stonkin in the region. And the farthest eastern sampling that is available in this dataset is the Cook Island sample. So our first admixture plots tells us that Easter Islanders here have actually their own component that is not shared within other Polynesian areas, but the only sample that could have something in common is precisely these Polynesian samples on the first block of the Polynesian, which are the Cook Islands. So not surprisingly, because these are the closest from this particular available data set. But there's still a big gap between the Cook Islands and Easter Island. So what we wanted to do is fill that gap with all these samples and hopefully tell if they come from this particular island, from any of those particular islands. So that's how we started this big collaboration also with people that have been working in the region for many decades. And this goes back to Professor John Clegg from Oxford University, that has been doing a lot of hemoglobin studies in the region and has a huge collection of samples. And Erika Hagelberg as well, who has been working in Easter Island for many years. So we basically put together a collection of hundreds of samples from all these islands, covering all the way from Taiwan through the Philippines, Indonesia, and then remote Oceania, and then Polynesia, trying to really compare the affinities of these populations. Also, since we were working in South America, we have the opportunity to compare also with South American populations. So I think this is the very first trans-Pacific study that we have a common data set to both address the question of the origin of Easter Islanders from the Polynesia, but also the big, long-standing question of whether Polynesians made contact with Native Americans or not. So that's the data. We combine it. Well, this is the initial sampling. We have roughly almost 200 samples genotyped in this Axiom Latino array, because that's what the platform that we studied. We initiated the genotyping with the South American samples. That's basically the technical reason why we stick to that. And then we combine it with reference panels as we did for other studies from both like continental references, African, Europeans, and Asians, but also Native Americans and other parts of the Pacific, like I show you on the map. So this is kind of a first look of the admixture patterns of the region. As you can see, well, the biggest pie chart is Easter Island for sure, because that's proportional to sample size. But as you can see clearly, well, there's a very clear pattern of this blue Polynesian ancestry coming all the way from Taiwan, very clearly distinguished from this Melanesian component that is not shared in most of the Polynesian islands, but rather they show an admixture pattern between this orange component, which is European, and that pink component, which is Native American. So interestingly, in Easter Island, there's a big proportion of that Native American component, but most of it comes actually from recent times, because as you know, and like I said, this is part of Chilean territory recently, so most of that is due to recent migrants that already have a Latino profile in terms of European, Native American, and recently some Polynesian as well. So it's not that easy really to disentangle where the Native American component comes from, either from recent times, or this could be a signature of an old contact from the Americas, which is something that we're really working on right now. So just to tell you that, to answer the question of where these Easter Islanders come from, we're applying two different approaches. And one of them is basically just reusing the long-standing statistics that have been widely used for many purposes in population genetics, the F statistics. So we're using an F2 version of it. It's just looking at similarities, genetic similarities between pairs of populations. So that gives us which is the closest related population from each group. But then additionally to that, we are doing a PSI statistic, which basically measures the change in the minor allele frequency between two populations. So when there's a bottleneck, for example, as in the case of Polynesian region, where you have a serial founder effect, so you can expect that some alleles will be lost, right? So by checking on those alleles that have been lost and the minor allele frequency of those that remain that have not been lost, you can detect which one is the parent population versus the child population. So if F2 gives us the closest pair, then PSI gives us the directionality of which is giving rise to which other one. So thanks to the combination of both, we can reconstruct the settlement of the whole region, thanks to the fact that these populations have been really isolated for many, many thousands of years. So after initial settling, these patterns have been really untouched, and this is the perfect setup for us to really detect even fine-scale differences between populations, because remember, this is also largely related among them, because they all come from the same expansion. So this is also a fine tuning between finding these small differences. But we have been able to be sensitive with this method, and I have to say also we have an ancestry-specific version of this, because remember, there's a lot of admixture, so we don't want to be influenced by this amount of European or Native American mixture, but this is ancestry-specific to the Polynesian portion of the genomes. So thanks to the combination of these two statistics, we've been able to do this graph, which summarizes basically kind of a cartoon version of the demographic history of the Pacific. We see that Samoa and Tonga are basically the root base of this Austronesian expansion. Then the dispersion hub, if you want to call it, is kind of the Cook Islands, where all the other islands, are coming from. So interestingly, Easter Island here, at least so far, our strongest signal is coming from a region that is combined either from the Marquesas Island in the north and these samples from Tahiti and some central part of the Tuamotu archipelago. So one of the other hypotheses where, remember the initial map, was that maybe they come from the Austral Islands like Rapa. Remember that Rapa, which is an Austral island, has a very similar name as the native name for Easter Island, which is Rapa Nui. So, for example, it could have been Rapa, which is not the case. Could be the Marquesas, because also if you look at the map of the Marquesas, many of the places are named Hiva Iti, Hiva Otu, and one of the legends at the origins of Easter Islanders is that their settler, embarked in a boat from Hiva. So, that's another cultural connection that might be the case. So we're finding some genetic support to some of those hypotheses, and we're still working on which is, at a finer scale, which is the particular island that gave rise to them. But this was just to give you an idea of what kind of questions we can answer with these population demography approaches and using genomic ancestry-specific tools. The other thing that we have, throughout this project developed in the lab is that not only we are giving back results to individual participants, but now we have the pipeline to do the same thing to any person from Latin America that wants to know more about their ancestry, right? So we're launching a service that is called actually Ancestry Service for Latin America. And here I'm just giving an example of a participant that can, give us samples to the lab, and we give back this ancestry proportion not only globally, but also like this local ancestry estimation to see where specifically in the world or in Latin America this person is coming from. So let me end with some, advertise about this service if you want to know more. And we have panels that are specifically enriched for the diversity of Latino populations and the Pacific. So this is the webpage where we can find out more about it. And with that, I will thank all the people that have been contributing to many of the projects in the lab. Neighboring institutions also have been developing a lot of genomic resources. We have joint lab meetings with them, and many people, like people here from ASU, Maria, that we're doing really great science even with limited resources. So with that, thank you very much.
Are you able to get any estimates on the timing of the historic ones, but for the Easter Island and the Polynesians and so on the timing of the dispersals and migrations?
Yes, excellent question. Normally we couldn't do that with snip array data, but something I did not mention is that we are developing yet another new method that is actually based on the decay of IBD, so segments that are similar between them, so identity by descent segments. There's also an exponential decay that we can expect if two populations mix like any X given time ago, you expect the decay to be of a given shape, right? So following that for each pair of populations and knowing on the background the graph that we obtain from which one gave rise to another, we can see just based on the decay how many generations were needed to explain that decay and based on IBD. So we are now mapping, we're now adding dates to the map of this different split pattern in the area. So that's really nice because that has not been possible to do so far with snip array data. But IBD is really powerful. IBD tell you about the recent history of populations, and it's a very strong signal about relationships in the recent time between families. And this basically is a huge Polynesian family that colonized the whole Pacific. So we can apply this particular method to estimate when that happened. So I'm going to tell you just very briefly, it really coincides with archaeological records. So that's really nice. So everything matches very well. That around the Cook Islands, all of the dates match similarly. And this is not really a long time ago. Again, this goes back to a very few thousands of years. And then you have, very single pulses out of that hub to colonize, for example, the long distant islands like Easter Island in 1,000 years ago, more or less, if I remember correctly. Any other question?
Thank you so much. This was fascinating. Could you tell us in general, what are the ethical issues that have been discussed? What are the ethical issues that you possibly foresee in the future for individuals, for populations, and so forth? I mean, just where has that conversation taken you thus far?
Right. No, that's a great point, because one of the things that we have been really careful about is to be ethical in every single sampling and field work that we have been doing for the past 10 years, like not only this Polynesian latest effort, but also throughout Latin America, to really have an engagement with the communities to do research only if they're really truly interested in that. Not like, hey, I'm just a researcher, that would be really cool to have a Nature paper, do you want to give a sample, right? So it's like, do you really want to understand more about your own history, and do you think that genetics could help you to do that? Because many people has a lot of fears, and with good reasons, that information may be, misused, or they don't really see a benefit that is going to be in the long term crystallized for them. And then in that, I give them that, because really genetics is not going to solve their immediate problems or health or anything like that. It's more like in the long term, we have to work with them to make them understand that this is a long time effort, that if we work with the community, we can understand more about them and eventually be benefit by the genomic advances that may be available in the next maybe 5-10 years. So that's not an easy thing to do when they are really desperate for another kind of help, right? But just giving them these things like salt or like building something or curing someone is like could be a more like immediate benefit, but I think it's more important to have like a long-term relationship of longitudinal relationships of projects that really stand longer for the community to understand the benefits of research. And unfortunately, not always is done like that. So because of other efforts that have not done well, we also go to the community and they have the doors already closed, right? Because in general, for them, it's the same thing if they come from the UK or Mexico or the neighboring lab. For them, they're all foreigners that are not from their community, right? So it's really important to understand what their needs are. And if they're really willing to participate, then we're happy to work together, right? So it's a very important point to really be careful about. Yes?
I'm curious how much information you have on just the basic descriptions of the individuals, their anthropometrics, their demography, and other components like their height, weight, and just basic health statistics on them?
It varies a lot because the majority of our projects come from different samples in different times, right? So back in the days, were mostly population samples that we had like no phenotypes at all from them, or in some cases just very few like anthropometrics, very basic stuff. In the more recent collections, like for example, the Biobank collection that I told you about from Mexico, it has a whole host of different metrics on health, on demography, so that we are hopeful that with that we will be able to do a better correlation with phenotype between genotype.
Do you have barriers to accessing the anthropometric information and or any of the other clinical information? Because I know in many of these registries, one of the over and above the challenge of collecting the information is then actually getting access to it or to be able to use it in the genomic context.
Right. Yeah, you're totally right. The most challenging part is maybe not the genetics, but the field work and collecting the right phenotype data. I agree with that. Yeah.
Any other questions? Well, with that, let's thank Andres again.
Thank you.